complicated slide, the opisthecons. So it's not a familiar term, but if you look up here, if you look at the black triangle on the top, those are metazoans, which are the animals, the multicellular animals. And they are part of that light blue group of organisms, all of the rest of which are unicellular. And we have some living unicellular versions of this. They're kind of cousins to us. Even though we're multicellular, they're unicellular, we have the same genes. Then there are fungi, and together with the metazoan, their cousins, and the fungi, we have opisthecons. So there's a wide group of organisms that we are part of. Now, all cells, whether they're opisthecons or other kinds of cells, they can all do the same things, unicellular organisms. They can switch between cell types. Bacteria can do this, and amoeba can do this. Cell surface variation, cell surface polarization. Cells become different one end from the other. Cell shape polarization. Cells become a different shape from the cell they originated from. They can secrete and release materials, they can secrete and bind materials, and they can oscillate from one state to another. You don't have to be a multicellular animal or embryo or plant to do these things. All cells can do this. So here, I showed some examples of opisthecons, and we have some present-day examples, and they're the cousins of us. Present-day, and they include fungi too. Present-day unicellular opisthecons contain genes whose products, and I give some examples here, mediate cell-cell interactions in animal embryos. They are collectively called the developmental genetic toolkit. This implies that these genes evolved for different unicellular functions and that their products were recruited to novel functions in the multicellular context. So what am I saying here? There were these very ancient single-celled organisms. They had these genes. The genes were not involved in embryogenesis because the cells didn't make embryos. They evolved to stick them to surfaces to allow them to catch prey. And then they found themselves in clusters of cells, and these genes started doing new things in those clusters of cells. So how do you get clusters of cells? If cells are adhesive, and there are some very ancient adhesive proteins, they didn't adhere cell to cell, but they adhered cell to surfaces. But under some conditions, like high calcium, for example, these proteins become adhesive, and you can get a transition from multicellularity, from single cells to multi-cells. And you can go backwards, too. And when you go backwards, it's called epithelial mesenchymal transformation. So that's the simplest physically mediated process that cells can do to make multicellular organisms. And if we look, the central panel with the yellow figures, those are about between 600 and 700 million years old. They're ancient forms that are found in the fossil record. And these were present way back before there were complicated bodies. There were these clusters. On the left, we see clusters of cells in modern-day embryos, and at the bottom, we see clusters that are made by subdivision of a large cell, as Ray Keller showed us happens in the frog egg. And the toolkit molecules that are involved here are things like cadherins and galectins and other lectins. They're just sticky molecules. Now, gastrulation, which we heard of, is a collection of processes by which animal embryos become multi-layered, establishing their body plans. Okay, and they have different names. In fish, epiboly is typical. Invagination occurs in frogs, and so on. Now, gastrulation can involve engulfment-like tissue rearrangement. But we know, remember I showed you those two liquids where one engulfed the other, the oil and the water? You could shake up the liquids. You do that when you make salad dressing, and then it separates out. So we find that with cells. If we take cells from different parts of the embryo and we mix them together, they will sort out and they'll form an equilibrium where one is engulfing the other. Or if you fuse the fragments of the same tissue, one is more cohesive than the other in general, and you get spreading of one over the other, and you get the same endpoint. So this is a physically ordained endpoint because of the nature of the cells and the tissues. Gastrulation often involves lumen formation. Lumens are interior cavities, and we can see that in sea urchins. It starts out as a cluster of cells, but then it becomes hollow. Well, if you have cells that are polar, that is, one end of them is less sticky than all the other sides of them, and the cells move around, you get a situation where you get a cavity inside because all the non-sticky surfaces don't find any partners. All the sticky surfaces stick to other sticky surfaces. So you get this spontaneously also by physical effects acting on cells that are polarized in a cell surface fashion. Gastrulation can involve buckling-like movements of tissue sheets. That is, you know that if you take a stiff sheet and you compress it, it will buckle one way or the other. It'll push out or push in. And you can see that in this fruit fly embryo. The cells buckle. Not the cells buckle, but the tissue layer made up of the cells buckles. And you can understand this on a physical basis as well. We heard some discussion of this in our symposium. If you have an epithelium, a tissue layer, and it has an underlying substratum which is a little different, it will buckle outward or buckle inward. It could become thicker, forming a placode. It can form branch structures. But this is understandable on the basis of the physics of sheets. And we can actually look at the elephant skin on the right and drying paint. And believe it or not, the physical processes that give you these similar patterns are not that different from each other. The flat configuration becomes unstable, and you get ridges forming because the paint is drying unevenly, or the tissue is buckling unevenly because of its underlying substratum or other properties. So if we want to understand the origins of gastrulation, we can consider differential interfacial tension, cell polarity, epithelial folding, and epithelial mesenchymal transition or transformation. And you can get all the body plans. You don't really need a lot of gradual evolution, and you don't need a lot of genetic change to get this. If you have the genes present, you can get one or another of these morphologies, and it does it by harnessing physics. Now, vertebrate and insect embryos typically undergo elongation, and we heard a lot about that in the earlier talk. And if we look at the mechanism that Ray Keller discovered and described to us of intercalation and convergence and extension, we can understand how we can get elongation. And again, it depends on a physical property of the cell, its ability to undergo planar polarization, and it has adhesive properties or differential adhesive properties, and you can write physical computational models to generate this behavior from these simple physical properties exhibited in a multicellular aggregate. Now here I'm just showing diffusion, which is a very simple physical process. If a cluster of particles starts out in one corner of a glass of water or a liquid, it can spread out and create homogeneity, or it can spread partially and create gradients. And the gradients can be used as information in multicellular organisms. You'd never know this by looking at a single cell releasing a material. It just releases it. But if it does it inside an embryo, you can get gradients. And here's a little more detail on that somite forming mechanism employing oscillations and gradients. And this is how this occurs. And you can see the somites lined up in a surface image of an embryo, or it's a drawing of one on the left. And you can see the mechanism as it plays out on the right. And there are toolkit molecules that were carried over from the ancient opisthecons that mobilize these different physical forces. So basically, if you combine physics and genetics, if you have the right genes present, and there's not that many of them, there's maybe about 24 of these toolkit genes that are involved in cell interaction, you get these different forms that can be used in various combinations. I'll just point out, look at the middle one. It is formed by something called lateral inhibition. So if all these cells have the same genomes, why is it that they're not all doing the same thing? And there is a little circuit that is almost always involving the notch pathway that we heard about earlier that causes cells to tell the cell next to them, don't do what I'm doing. So that's just one example. And that is used in other things like the stripe forming mechanism that we see in somite formation, and also fingers. It's now known that the digits of the hand and the toes of the feet are formed by these Turing type of mechanisms that involve lateral inhibition and diffusion. Now, on the top, we're looking at a single-celled example of a modern-day opisthecon, a choanozoan, and that has a lot of the genes that eventually became the embryology genes. And I just gave some names of them there. And then those are just single-celled. When we got slightly more complex forms because of what's called homophilic adhesion, where the cells started to stick to each other, you got simple forms like placozoans or sponges, the periphery. They don't have all of the morphological complexity of modern-day forms because they don't have all the genes that are present in this toolkit. Those genes came in somewhat later. Some of them were imported from fungi and from other organisms. So basically, you got more and more complex as you acquired more of these genes, but the genes didn't just act by being genes producing new proteins. Those new proteins, finding themselves in a multicellular context, mobilized some of these mesoscale physical processes that were not available before to a cluster of cells, and then the cluster of cells became morphologically more complex. So now we're down to insects and chordates, and sad to say insects are about as complex as we are, so I can't distinguish us from them on this basis. Now, the modern synthesis, I'm coming back to Ernst Mayr, who was considered one of the founding figures of the modern synthesis. And I'm using him as what we call a whipping boy again, because I'll describe his viewpoint about what Darwin's advance was and the theory of natural selection. And this was before embryology and the physics of form got incorporated into the understanding of evolution. He said, nothing strengthened the theory of natural selection as much as the refutation, one by one, of all the competing theories, such as saltationism, that is the ability of organisms to jump suddenly from one form to another, orthogenesis, That is the predictability of form. Now, as I showed you, you can actually predict forms if you know the physics, because each kind of mesoscale physics has its own characteristic morphological outcomes. So Meyer said that it was necessary to reject orthogenesis and the inheritance of acquired characteristics. Okay, he said that in 1982. We now know that each one of these factors played important roles in the evolution of form. So toward a new evolutionary synthesis, we can assert from what we know now, animal body plans originated from the combined actions of physical processes mobilized in multicellular aggregates or clusters by ancient gene products. And those gene products took on new functions in the multicellular context. The evolutionarily earliest developmental mechanisms, being based on physicogenetic processes that were typically self-organizing and often nonlinear, readily produced abrupt saltational changes in organismal form. The physicogenetic determinants of multicellular form ensure that body plans of animals and plants have preferred stereotypical morphological motifs, multi-layering, segments, cavities, so on. That is, generation of form is orthogenic. The outcomes of physicogenetic determinants of form are dependent on and sensitive to the external physical and chemical environment. Since physical laws are passed on along with genes and determine how genes are expressed, acquired morphological phenotypes can be inherited. Okay, now we see that less in modern day organisms because there are so many backup mechanisms and redundancies so that embryos tend to look like they're programmed to go in a certain direction. But if you think about the most ancient embryos, they were much more susceptible to physical changes in the environment than modern day embryos. And even modern day embryos have this flexibility. There was an experiment that was done in the 1950s by Anne McLaren, a famous geneticist. She swapped mouse embryos between different strains of mother mice. And some mice embryos had 6 vertebrae in their necks and some had 5. And she could get the morphological phenotype to change by changing the gestational environment. So even modern day organisms are sensitive to this influence. Okay, thought I had a slide with my colleagues, but maybe not. Okay, so that's it. Thank you. Let me just close this.
Abrimos la sesión de preguntas.
¿Aló? Sí. Quería preguntar si las fuerzas físicas que van modelando la forma de los planes corporales parecieran ser ilimitadas. ¿Cómo se explica que exista un número tan limitado de planes corporales, como lo mencionó al principio?
Well, yes, I think that the idea that they're unlimited is really not a valid idea. I think that we find similar motifs appearing again and again in the history of life. In fact, insects have segments and vertebrates have segments, but their common ancestor didn't have segments. The same genes were recruited independently to make segments in both lineages, but this is this is an example of recurrence of a morphotype. Not the morphospace of the animals is not unlimited and you can't go continuously from one form to another. You make jumps between them.
Hola. Sí, yo tengo una pregunta acerca de la función. Explicaba en sus primeras diapositivas que en definitiva los genes ancestrales estaban presentes en las formas más preliminares, en las formas inclusive las unicelulares, y que de alguna manera esos genes estando presentes ahí cumplían funciones distintas que lo que podían cumplir después durante la organización más multicelular. Mi pregunta es acerca de cómo eventualmente puede evolucionar la funcionalidad de un gen determinado, si eso está establecido de alguna manera o si se sabe y qué tanto la física de alguna manera es capaz de modular también la funcionalidad de ciertas proteínas.
So, the functionality is really, the form precedes the function. So if you have an adhesive protein on a cell, and it actually evolved to enable the cell to stick to a prey or stick to a surface, and then the local environment changes in its ion content, and the cells then become sticky to each other, well, you could say the new function is to stick cells to each other. I mean, it's just a matter of definition. The question is, how does that new form establish itself in the environment and become functional? And here is a new branch of evolutionary theory, which I did not refer to, but is equally important, and that's the idea of niche construction or niche selection. And the idea there is that organisms with new types of forms can actually actively seek out places in the world where they are functional, where they can survive. So unlike the Darwinian model where organisms are passive recipients of their environment and stand or fall depending on how well they fit pre-existing niches, the new idea about niche selection is that organisms are active and find places where they can survive by means of their resourcefulness.
Tengo una pregunta. ¿Cuál es la fuerza inicial que hizo que piensas que los organismos unicelulares llegasen a producir estos organismos multicelulares? ¿Fue algo más bien al azar? ¿Es algo dirigido?
Well, it's known that cadherins, which are the major adhesive proteins of multicellular animals, existed in single-celled ancestors, but they did not mediate attachment of cells to each other, or they didn't mediate multi-layering of clusters of cells. But it's known that in the geological era that accompanied the rise of multicellularity, there was a change in the ion content of the oceans, and calcium rose during that period. Now we know that cadherins are calcium-dependent cell-cell adhesion proteins. Without calcium, they don't mediate the sticking of cells to one another. So I wouldn't quite call it random, but it was an environmental effect that possibly mediated the attachment of cells where that didn't exist before. Thank you.
Unas últimas palabras antes de finalizar. Primero, queremos agradecer la voluntad que tuvieron Ray Keller y Stuart Newman de acceder a estas conferencias al público. Esto no es algo que comúnmente se haga y nosotros estamos realmente agradecidos que ellos hayan aceptado esta invitación. Además, quiero agradecer a algunas personas que tuvieron un papel muy importante en organizar este evento. Primero a Guido Gerardi como presidente de la Comisión de Desafíos del Futuro del Senado de Chile por permitir que esto ocurriera acá. A Carola Muñoz, asesora del Consejo de Futuro y parte de la organización del evento. A Antonella Sanguinetti, encargada de comunicación del Instituto de Neurociencias Biomédicas. Y en especial a Paola Cañón, directora ejecutiva del Instituto de Neurociencias Biomédicas, que ha tenido una participación muy importante en organizar este evento. Un aplauso para ellos, por favor. Entonces, damos por finalizado este evento y quiero agradecer a ustedes el haber venido acá, a los estudiantes de enseñanza media, y espero que este evento les haya logrado despertar algún interés en esta temática que no es muy común y no muy abordada en términos de extensión. Bueno, hasta luego. Muchas gracias.